Welcome. I'm Tilly Bingham, VP of Science at Concept Life Sciences. Thanks for joining today's webinar, where two of our scientists, Dr. Thomas Pesno and Dr. Max Ahmed, are going to talk to you about multicellular tissue models and methods to identify tool compounds for interrogation of novel targets. Before I hand over to my colleagues, I'd like to very briefly introduce you to our company. Concept Life Sciences is a UK-based CRO serving the pharmaceutical, biotech and agrochemical sectors. We have an established track record of delivering complex and specialist programs for our customers, and we have particular expertise in the area of neuroscience discovery research. Neuroscience research has a range of unique challenges, not least in terms of target validation and human translation, as well as the unique requirements for the therapeutic agents to cross the blood-brain barrier. And these are themes that our two speakers are going to touch on today. Our talk today will be in two halves. The first from Max Ahmed in our neuroscience biology team, followed by Tom Pesno from our medicinal chemistry team. Both speakers will be covering strategies for the early stages of discovery research in target validation and hit discovery. It's critical to get this phase right as it provides the foundation for all the research that follows. Although we're talking about the early stages here, it's important to emphasize that the biology models established during this target validation stage are often the ones which are key throughout the lifetime of your program, and are often also used to select the final candidate. As such, it's critical to select the best, most relevant models which can be reliably used for candidate decision-making later on. Similarly, the selection of the right kind of hit finding strategy is critical to provide the best quality hits for elaboration, bearing in mind there are additional molecular design constraints for neuroscience projects. It's important to ensure that the design strategies and hit finding are maximized to provide high quality CNS-like lead compounds. Both speakers today will cover these areas and highlight how the right decisions at this early stage will ensure you establish a sound foundation for your project. Without further ado, I'd like to hand over to our first speaker, Max.
Hi there, my name's Max Ahmed, and I'm going to be discussing some of the multicellular tissue models that we've developed at Concept Life Sciences to support drug discovery projects. As I'm sure many of you are fully aware, CNS drug discovery is not for the faint-hearted. The high attrition rate is really reflective of the complexity of the human brain. And because of this, there is an increasing interest in trying to understand the cellular and molecular mechanisms that underpin a lot of pathology and repair mechanisms in the brain. To date, most of the research has focused on neurons as a principal cell that executes neural functions, and these have long dominated the ideas underpinning most disease pathology. However, I think it's increasingly recognized that neurons don't exist in isolation. In fact, they're surrounded by a complex environment of multiple cells, and it's quite a heterogeneous, spatially constrained structure. And you can see here in the diagram that the neurons form cell-to-cell connections with other neurons, and this is what facilitates the electrical conduction and neuronal signals in the brain at each of these synapse points. The axon of the neuron has these lipid layers called myelin generated by the oligodendrocyte, and these myelin layers tend to regulate electrical conduction along the axon. And along with the oligodendrocyte, you also have astrocytes, which form a bridge between the blood vessels and the neurons, passing on signals. And amongst all this, you have the innate immune system, cells of the innate immune system, the microglia, which regulate many of these functions. And none of these cell types act either alone, but in fact, they are all interconnected and function together. And we know that in a number of pathological conditions, both astrocytes and oligodendrocytes can become immunocompetent, and what makes the situation even more complex is that in pathological conditions, we know that the peripheral immune system can infiltrate the CNS and play a regulatory role there as well. And so what you have is a really quite a complex multicellular environment that is dependent not just on the different cell types, but how these are organized, maintained, and structurally connected. And so to be able to interrogate CNS biology, we really need to be able to recreate this model with greater fidelity in the lab. The disease that best characterizes this multicellular nature of CNS pathology is probably multiple sclerosis. In MS, circulating immune cells infiltrate the CNS and start attacking the myelin that coats the neuronal axons. And as myelin is degenerated, this disrupts the nerve signals that propagate in the brain, and you're left with an exposed axon. And unless that exposed axon is repaired by remyelination, you eventually end up with degeneration of the neuron, resulting in progressive disability. So as I mentioned, remyelination is possible. There is a population of tissue-resident progenitor cells, oligodendrocyte progenitor cells, OPCs, that are able to differentiate into newly myelinating oligodendrocytes and repair this exposed axon. However, the mechanisms that underpin that repair process are poorly understood, and indeed the mechanisms that drive the initial degeneration are not that well understood either, and this really motivates us to develop better tools to be able to understand this process. Now, we can use standard tissue cultural techniques to culture these oligodendrocyte progenitor cells, and we can also promote the differentiation of these OPCs into mature oligodendrocytes that deposit myelin. However, as you can see in this image, the ability to produce and deposit myelin in a flat dish is not the same as the in vivo environment, where the cell, the oligodendrocyte, makes contact with up to 50 axons independently and is then able to wrap these axons in this concentric myelin layer. And we know that this process is regulated via multiple different cell types, microglia, astrocytes, and even the underlying axons play a role in the development of these myelin sheaths. And so to better understand this process, we really need to develop a platform that has these multiple cell types and that three-dimensionality to allow myelination and wrapping to occur. And it's for this reason that at Concept Life Sciences we've developed this organotypic slice model. And in this model we're able to keep all the supporting cells viable, and you can see that here where we've labelled the microglia with CD68 and IBA1. The astrocytes are labelled with GFAP and are distributed throughout the tissue, and you can see here the population of oligodendroglia labelled with NG2 and OLEG2. And what's more, we have robust myelination in this system, where not only is the lipid layer deposited, but you can see in this higher MAG image that the oligodendrocytes are able to make contact with the axon and really wrap them like you would expect in a native brain environment. The main advantage of the system is really that it maintains all the relevant cells in their appropriate structure and organization, so providing that form and function of the brain, but remaining quite tractable and easy to manipulate. The complexity of the system renders it unsuitable for high throughput programs, but for low throughput validation of target or candidate molecules, it's a very useful translational link between those really quite simple in vitro studies with dissociated cells and the far more complex full in vivo studies. The readouts can range from image-based immunohistochemical techniques right the way through to gene expression via qPCR or RNA sequencing, and the platform is flexible enough that we can really tailor the assay setup depending on the scientific question that's being asked and meet any individual experimental need. So I provided here one example of a study we performed using this system, where the aim was to determine the potency of a tool compound using qPCR to measure the expression of a downstream target. Now, prior studies showed that there was a mismatch in the in vitro data generated from dissociated cells and the in vivo pharmacology. And so the challenge was to determine whether these organotypic slice models better recapitulate the in vivo data. And so we generated the slices by harvesting brains from the neonatal mice aged between P0 and P2. We were sectioned about 300 micron thick coronal sections of the brain and kept the cortices in culture and treated them with the compound in a dose-dependent manner. We then collected the slices, isolated RNA, and performed qPCR for the target, for the downstream target, and the data is shown here on the right-hand side. And relative to the vehicle, we were able to induce expression of the downstream target up to 15-fold at higher concentrations, and so determine a IC50 of the tool compound that better reflected the in vivo environment. And you can see here that each data point is an independent animal. It's a slice generated from an independent mouse. And so, as I referred to earlier, there is increased variability. However, the data generated was more reflective of what's found in vivo compared to the in vitro dissociated cell system. And here I'm sharing a second study we've performed using this platform, where the aim was to determine the efficacy of any potential biological target to drive remyelination. So again, we started by harvesting the brains of neonatal mice pups, which we sectioned and collected the cortices and kept them in culture. And as I showed in slide 5, these slices are able to myelinate quite robustly. And once they do myelinate, we can apply a toxin to strip the myelin off the axons, leaving behind the bare axons, thereby creating a MS-like lesion as a disease model. These slices can then be treated with either a tool compound or a biologic, a recombinant protein or such, allowing us to determine what role they play in driving the repair process. And at the end of the study, we fix the slices, stain with antibodies against MBP neurofilament, and expose to confocal imaging and automated image analysis pipelines. And an example of the data is shown here on the right hand side, where the axons are highlighted in green with neurofilament staining, and the newly generated myelin is shown here in red MBP. And what we do is quantify the length of the axons and the co-localized MBP staining and determine a ratio of remyelination. And you can see here an example of the data set where, compared to vehicle, we're able to induce a 2-fold increase in remyelination with a control, and thereby you have a platform where you can essentially validate your biological target in quite a controlled environment compared to a full in vivo setting. So in summary, I hope I've been able to convince you of the need for these multicellular 3-dimensional models to study this complex biology in CNS drug discovery, and that the platform that we have developed here at Concept is quite a useful link between these simpler in vitro assays making use of dissociated cells and the much more complex in vivo assays that I'm sure we're all familiar with. But of course, having validated any potential target, we need to develop novel tool compounds to interrogate the potential target. And here at Concept Life Sciences, we work closely with medicinal chemists, and my colleague Tom Pesno will now take you through one approach, a fragment-based drug discovery approach that we have for identifying novel CNS matter. Thank you.
Thank you, Max. So I would like now to use the last few minutes of this presentation discussing hate identification and how fragment screening can be a very useful tool for the identification of hit matter for CNS therapeutics. There are a range of strategies available in the medicinal chemistry toolbox to identify hits for drug discovery campaigns, starting from literature-based approaches such as scaffold hopping, all the way to the screening approaches, so HTS and fragment screening. And there are a range of questions that we need to ask ourselves when initiating a hit identification campaign. First of all, how much literature data there is available, but also what we need to understand the biological target class and if there's any restrictions associated with that. And finally, the therapeutic area is especially relevant for CNS targets. As is stated here, brain penetrant molecules tend to be significantly smaller than peripherally restricted molecules and significantly less polar. And so these differences need to be taken into consideration when selecting the hit identification campaign. In that instance, fragment screening seems like a very useful strategy for CNS drug targets. The fragment-based drug design rationale states that drug molecules can be considered as a combination of smaller fragments which can then be bolted together to generate the drug molecule. But these fragments can be screened individually to generate hits. And this is essentially what is fragment screening. We take a fragment library of about 1,000 molecules. By definition, these are fragments, so they are very small molecules, and they are screened against the target of interest to identify hits. And these hits can then be progressed into lead and clinical candidates using various approaches such as fragment growth or fragment merging. Obviously, hits identified through fragment-based screening will be small molecules, and so it gives us a much greater chance of improving those hits into the small CNS drug-like targets to generate clinical candidates, as opposed to high-throughput screening where the start point, the hit, would be a much bigger molecule. But aside from the size, there are a range of advantages in using fragment screening over high-throughput screening. First of all, operationally, it is significantly quicker and cheaper to run a fragment screening campaign. In terms of hit rates, the hit rate is orders of magnitude higher in fragment screening than it is for high-throughput screening, and that's essentially associated with the efficient sampling of the chemical universe when screening much smaller molecules. And fragment screening has been a successful approach for a range of difficult to drug targets. The question really, and especially for the CNS, is fragments are obviously small molecules that will give us small hits, but how small can we go in terms of fragment screening? And this will be hinged on our ability to detect very weak binding events, simply because the smaller the molecules, the weaker the binding event associated with it. And interestingly, at the same time as fragment-based drug design has emerged as a strategy for drug discovery, so did a range of biophysical technologies. And these biophysical technologies allow for the detection of very, very weak binding events, such as high as 10 millimolar. And these binding events these detection methods can then allow for the detection of binding for the very smallest of fragments. Now, this biophysical technology so far has not really been maximized in the commercial fragment collections that typically are in the bigger or higher range of the fragment landscape. So we thought that we should actually try to maximize the capability of biophysical screening by designing a fragment collection that efficiently samples the very limits of the biophysically accessible chemical universe, which we call this biophysical sweet spots. And in turn, we anticipate that such a library would have a very much improved screening hit rate because of a more efficient sampling of the chemical universe. And also we would detect much leaner hits and so much higher quality hits. So we set out at Concept Life Sciences to design such a fragment collection using our internal compound collection of 55,000 molecules. We carried out physicochemical filtration, targeting these biophysical sweet spots in terms of size and lipophilicity. We also carried out very stringent clustering and quality control to ensure that we had a maximal sampling of the chemical universe and the very highest quality fragments. The library which we designed contains about 1,100 compounds, and we compared the properties, the physicochemical properties of our library in blue to that of typical commercial fragment collections in green and fragment hits in red. And as you can see, our fragment library does not only fit within these biophysical sweet spots, which we hope will maximize our ability to detect lean hits, but we also address some of the differences between typical fragment collections and what typical fragment hits look like, which have been reported over the past 10 years. So not only in terms of hydrogen bond acceptors, in terms of rotatable bonds and fraction sp3, so 3Dness, as well as obviously the size. In all categories, we have a very good match of properties between our fragment collection and that of typical fragment hits. So we now combine this, at Concept Life Sciences, combine this state-of-the-art fragment collection with our expertise and technology in biophysical screening assays to identify the very highest quality fragment hits for drug discovery campaigns, whether it's for the central nervous system or for other therapeutic areas. And hits, the quality of the hits is really critical to any drug discovery campaign. And I would encourage you to take a look tomorrow at Natalia Markova's presentation of the fragment screening of PARP 15, and it'll really highlight to you that attrition is significant during a hit confirmation in fragment screening, and concerted approach in terms of a range of orthogonal strategies need to be used to ensure that we have the highest quality hits. And obviously in this instance, it is especially important to come up with or to screen a very high quality fragment collection to minimize attrition during the hit confirmation and hit lead campaigns later on.
Thanks, Tom. Hopefully we've convinced you that taking time to establish a sound foundation for your project will reap dividends downstream. We've only had a chance to present a fraction of the neuroscience work we do in this short seminar today, but if you'd like to know more, we'd be delighted to discuss with you how we can help advance your programme.